Okay, so welcome back to our second session for biochemistry. We are going to be looking at the metabolic pathways uh, today. We'll start with the amino acids and proteins, then talk about carbohydrates, and finally we'll talk a little bit about lipids. At the end, we'll mention briefly about some of the vitamins. Okay, so let's start. Let's start. Okay. So first, the basic structure for amino acid. This is just for a recap, so you know what molecule are we talking about. So very basic structure. So in the structure, you have what is called the central carbon. We call it the alpha carbon. To this central carbon, there is the attachment of carboxylic group. So wherever the carboxylic group will be there, that carbon will be called the alpha carbon. Okay, and to the same carbon we will typically have the amino group. We will typically have the amino group at the same carbon. Okay, so we have the carboxylic group and the amino group. So that is amino acid. See, this is the amino and this is the acid part. So these are the two main components of the molecule called the amino acid. Very simple, very obvious. Okay, in addition to this, you will have one hydrogen at third valency. The fourth position is the one which shows the variation. So we have what we call the unique side chain. We have what we call the unique side chain R, and this R will be H in glycine. The simplest amino acid is glycine. So in glycine, this R will be equal to H. In others, the R will be different. So it is the R which is going to vary. It is the R which will vary in the different molecules. In the different amino acids, the R will vary. One small thing I would like to mention here. So where the carboxylic group is there, this is the alpha carbon. So the amino acid that you get is the alpha amino acid. Okay. On the other hand, if the amino group were to be attached, let's say here the amino group is there, then it is the alpha carbon. If the amino group is attached to the second one, I'm sorry, if it is attached to the second one, the second molecule, second carbon would be the beta carbon. And if the amino group is attached here, if the amino group is attached here, in that case we call it the beta amino acid. So we have the alpha amino acid. Commonly we talk about this alpha amino acid, but if the amino group is placed at the second carbon, then it will be the beta amino acid. Similarly, sometimes the carbon group may be attached at the third carbon. If it is attached to the third carbon, then obviously what it will be? It will be the gamma amino acid. So alpha amino acid is the common amino acid that we come across, but depending on position of the amino group, you can have alpha, beta, and the gamma amino acids. You can have the different types of amino acids. Okay. Second thing, when we talk about amino acids, we mentioned there are 20 amino acids. Why 20 amino acids? These are the amino acids for which the genetic code is there. So if you look at the sequence of nucleotides, the nucleotides will make the codon having 3 nucleotides, and this codon translates into the amino acid. These codons are getting translated only into the 20 fixed amino acids. So we talk about those 20 amino acids mainly. Please note, they are L-type alpha amino acid. They are L-type alpha amino acids. Some of them are dextrorotatory, some of them are levorotatory. But for dextro and levorotatory, we use the symbol plus and minus. This is not L. No, this is not true. L is for the stereoisomerism. Okay. Whereas when we talk about the optical activity, we call it dextro and levorotatory, for which we use the symbol plus and minus. For which we use the symbol plus and minus. Okay, so remember, some amino acids are dextrorotatory, some are levorotatory. Which amino acid doesn't have optical activity? The answer should be glycine. Glycine doesn't have optical activity. Okay, this is a very simple straightforward question which you have to remember. The simplest amino acid glycine does not have any optical activity. All other amino acids are going to have the optical activity. Now, whenever we talk about a group of molecules, we try to classify them. So, for amino acids also, we can have various types of classification. We can classify them based on whether they're acidic, whether they're basic, whether they're polar, whether they're non-polar, whether they're ketogenic, whether they're glucogenic. So, a lot of criteria for doing the classification is there. But we just mentioned the main difference among the amino acids is the R side chain. Okay, we said the main difference is the R side chain. So, what we do, we use the side chain for doing the classification. Okay, and for this you can see very simple. Some of the amino acids have only the aliphatic side chain, only the aliphatic side chain. This includes glycine, alanine, glycine, alanine, valine, leucine, and isoleucine. These are the five aliphatic amino acids: glycine, alanine, valine, leucine, isoleucine. We have written this here: glycine, alanine, valine, leucine, and isoleucine. In this, what you have to remember, the last three here in the side chain, there is branching also. So these three are also known as the branched amino acids. These are also known as the branched amino acid. Okay, so valine, leucine, and isoleucine are the branched amino acids. So whenever you're talking about the branched amino acid, it has to be one of these three. For example, when we talk about the condition called the maple syrup urine disease, where there is problem in metabolism of branched amino acid, so there one of these three has to be there in the option. If they ask which amino acid is elevated, so you have to remember it has to be a branched amino acid, and it has to be one of these three names. Okay, moving on to the second one, side chain containing hydroxylic groups. Okay, so here the names are serine, threonine, and tyrosine. The names are serine, threonine, and tyrosine. Okay, serine, threonine, and tyrosine, they will have the hydroxyl group in the side chain. Then we have side chain containing sulfur atom. I'm sure all of you know these two names, the side chain containing sulfur atoms. These are methionine and cysteine. Methionine and cysteine. These are the two amino acids having sulfur in the side chain. And then we have the side chain containing acidic groups or their amides. So what do we get here? First we get the aspartic acid. First we get aspartic acid, right? This aspartic acid can get converted into asparagine. Okay, so aspartic acid is the acidic one and the amide derivative is the asparagine. Similarly, you have the glutamic acid, and this glutamic acid will get converted into the glutamine. So these are the names that we have written here: the aspartic acid, asparagine, glutamic acid, and glutamine. Glutamic acid and glutamine, these are the amino acids either having the acidic group or the derivative of the acidic group, which is the asparagine and the glutamine. Then we have side chain containing, sorry for the disturbance. Okay, so side chain containing basic groups. So here what you have to remember, we have the histidine, lysine and arginine. Histidine, lysine and arginine. For this you can have the mnemonic HLA. Histidine, lysine and arginine. The mnemonic is HLA. And then the last important one containing aromatic rings. Last important one containing aromatic rings. So here you have to remember the names. Here you have to remember the names. Phenylalanine, tyrosine, tryptophan, and H is for the histidine. Okay, H is for histidine. So phenylalanine, histidine, tyrosine, and tryptophan. So these are the amino acids having the aromatic ring in the side chain. They have the aromatic ring in the side chain. And last, we have an amino acid which is commonly known as the amino acid. Amino acid which is known as the amino acid, the answer is proline. Okay. So you commonly get questions from the ring structure, which of the amino acids is basic, which of the amino acids is acidic. These are some of the important common questions. Which of the amino acids is branched. So once you know this classification, these questions become very simple, straightforward, one line questions. Okay, please don't make mistake in these questions. They will get you the easy 4 marks in the exam. Okay, moving on. Sometimes, not always, but sometimes you can get the question based on the metabolic end product of the amino acids. On the basis of this property, we have the ketogenic and the glucogenic amino acids. Now what you have to understand here, there are some amino acids which are both ketogenic and glucogenic. Some amino acids which are both ketogenic and glucogenic. So you can remember them by the mnemonic PIT. Okay, what is PIT? P, obviously you know, P is for phenylalanine, right? P is for the phenylalanine. I is for isoleucine. T is for tyrosine, and another T is for the tryptophan. So these are the 4 amino acids which are both ketogenic and glucogenic. The 4 amino acids which are both ketogenic and glucogenic. On the other hand, second important question which is asked is amino acids which are exclusively ketogenic. So there your answer should be lysine and leucine. Lysine and leucine are exclusively ketogenic amino acids. All others are exclusively glucogenic amino acids. All others are exclusively glucogenic amino acids. So I'll repeat, exclusively ketogenic. Exclusively ketogenic. Answer is lysine and leucine. Amino acids which are both ketogenic and glucogenic. Both ketogenic and glucogenic. Answer is phenylalanine, isoleucine, tyrosine, and tryptophan. You can remember them by the mnemonic PIT. How do you remember this PIT? This is the PIT of Brad Pitt. Pit of Brad Pitt. So now you are not going to forget that this pit is going to have the 2 T's. This is the pit of Brad Pitt. So remember it like this. This is the pit of Brad Pitt. Okay. So there are the two main methods for classifying the amino acids. First, based on the R side chain. Second, on the basis of metabolic end products, whether they are ketogenic or they are glucogenic. Rarely you may get questions on the nature, whether they are hydrophilic or hydrophobic. So please make note of these amino acids which are hydrophilic in nature: glycine, serine and threonine, cysteine, aspartic acid, asparagine, glutamic acid, glutamine, histidine, lysine, and arginine. Okay. In addition to this, the following amino acids would also be charged. The others are polar, but they are non-charged. These ones, the acidic and the basic amino acids, they are polar as well as they are having the charge. Okay, so in this way you can remember the hydrophilic and the hydrophobic amino acid. So the others would be hydrophobic. Okay, these are the only ones which will be hydrophilic in nature. Sometimes to this list you might add tyrosine. Remember, in tyrosine we have the hydroxyl group. I told you the hydroxyl group is there. So because of that, it has some hydrophilic nature, but at the same time it is aromatic in and the right side chain is aromatic, so which causes the hydrophobic nature. So it is a borderline hydrophilic. If you have to choose that, a lot of options are there and tyrosine where to put, so you may decide to put it in hydrophilic, but remember it is weakly hydrophilic because of the aromatic ring. It is weakly hydrophilic. Now, this 21st amino acid being asked again and again and again, you should know about this 21st amino acid. Number one, what is the name of this 21st amino acid? Okay, this question has come at least 3 times in the last 3 years in our exam. So you should know about this. It keeps coming up some very frequently. It will land up in the FMG exam, but it has also come in our written exam. So you should know about this. Selenocysteine. First point to remember is it gets added to the peptide chain during translation. See, it is written co-translational insertion. Okay, so it is not added as a result of post-translational modification, but during translation. That is what we are writing, co-translational insertion. It is added during the process of translation. Second, it is present in several mammalian enzymes, and this is the second question which is asked. Which of the enzymes are going to contain the selenocysteine? So some names which you have to remember, I'll write here. Some names which you must remember. First, glutathione peroxidase. Glutathione peroxidase. Second, thioredoxin reductase. Third, deiodinase. Third is deiodinase. Fourth important name is glucose 6 phosphate dehydrogenase, commonly known as G6PD. Commonly known as G6PD, the glucose 6 phosphate dehydrogenase. In addition to this, a lot of enzymes involved in the selenocysteine synthesis, selenocysteine synthase, etc., they also contain the selenocysteine. So if you remember these common names, then most of your work is done. Last point which you have to remember here is the codon. See, the codon for this is actually a stop codon, but it has a very special feature. The stop codon is followed by what is known as selenocysteine insertion sequence. It is followed by the selenocysteine insertion sequence. If this sequence is not there, you commonly write it as SECIS. If the sequence is not there, then the UGA will work as a stop codon. But if this sequence is there, then the UGA will code for the selenocysteine. Selenocysteine can get incorporated at this point. Is this point clear? Okay, great. Okay, okay, okay. I can see there is one question in our group. It was asked in the exam yesterday. I'll come to that question shortly. Okay, once when I'm taking the break, I'll take that question. Okay. Uh, because we're talking about 21st amino acid, please note down another name, just the name, nothing else, nothing else. Why nothing else? Because it is not found in humans. The another name is pyrrolysine. It is the 22nd amino acid. I'll not tell you anything else about this except that it is found only in the bacteria. It is found only in the bacteria. No other information is required at this point. Okay. So now we quickly go on to the metabolism, or you can say disposal of the amino group of the amino acid. Okay, we are looking at the disposal of the amino group of amino acid. This is the one from where we are going to get maximum questions in the amino acid for our exam. Okay, so this amino acid metabolism for the amino group is described in 4 stages. Stage 1, transamination. Stage 2 is oxidative deamination of glutamate. Stage 3 is ammonia transport, and stage 4 is the reactions of urea cycle. Okay, so we'll quickly look at each of these stages one by one and see what are the questions which they are asking from these stages. Starting with the, okay, this is again the diagrammatic representation. As you can see here, the transamination is happening, then the glutamate undergoes oxidative deamination, the ammonia is taken to the liver, and ultimately we have the urea cycle, which is stage 4. So in transamination, what you have to know first, what is the definition, how do you describe the transamination? So in transamination, the, if you look at the structure, like what is happening in the reaction, we have the amino acid and it interacts with a keto acid, right? We have the amino acid interacting with the keto acid. In this process, what happens, the amino acid transfers the amino nitrogen, it transfers the amino nitrogen, and the amino acid becomes the keto acid. I'll write it here, amino acid 1, keto acid 2. So the amino acid now becomes the keto acid. The keto acid receives the amino group, which is commonly the alpha ketoglutarate, and it becomes the amino acid. In this case, it becomes the glutamate. So you can see here, the alpha ketoglutarate, which is the keto acid, gets converted into the amino acid, which is the glutamate. So this essentially is a transamination. What you have to remember, the transamination reaction is freely reversible. Second, PLP means the vitamin B6 pyridoxal phosphate. PLP stands for the pyridoxal phosphate. PLP is required for the transamination, and some amino acids do not participate in transamination. This is another important point which you have to remember. These include the lysine, threonine, proline, and hydroxyproline. So what all did I tell you? First, what is the definition for transamination? Second, which cofactor is required, which vitamin is required, which form of the vitamin is required, that is second. Third, I told you this reaction is freely reversible. This reaction is freely reversible. And finally, I'm telling you that there are some amino acids which do not undergo the process of transamination, including glycine, threonine, proline, and hydroxyproline. Okay, so this is the same thing. You can see here the amino group is getting transferred to the keto acid, and keto acid becomes the amino acid. Okay. So now what has happened? All the amino group has gotten accumulated in the form of the glutamate. We told you recipient is the alpha ketoglutarate. It will take up the amino acid and it will get converted into the glutamate. So from this glutamate, we have to take out the ammonia in the free form. The process is known as the oxidative deamination of glutamate. Here, what we are going to do, we are going to release the amino group which is now attached to the alpha ketoglutarate. What happens? The glutamate is split into the ammonia and alpha ketoglutarate. See, when we are doing the transamination, in that case, the ammonia is not in the free form. But here, when we are doing the oxidative deamination, the ammonia that we get is in the free form. Okay, and for this purpose, any of the two, either NAD or NADP, any of the two can be used, resulting in formation of either NADH or the NADPH. Depending on which molecule is participating, we can either get the NADH or we can get the NADPH. Okay. Okay. What is important to note is, just like the transamination reaction, this reaction is also freely reversible. This reaction is also freely reversible, catalyzed by the glutamate dehydrogenase. Now, why is this information important, that this reaction is freely reversible? Very important question which is asked is, If there is hyperammonemia, because of any reason the ammonia levels will rise, then there is encephalopathy. What is the cause or what is the effect? So you have to remember here, when the ammonia levels will rise, obviously the backward reaction will become more favorable. When the ammonia level is high, or at one end the product is high, then the backward reaction will become more favorable. So this ammonia will start combining with the alpha ketoglutarate to regenerate the glutamate. But what happens in this process? The alpha ketoglutarate will get depleted. We don't have lots of alpha ketoglutarate. We only have lots of ammonia, right? So ammonia will start moving backwards, and alpha ketoglutarate will start getting depleted. It will start getting depleted. We have a little monkey here. A monkey has come in. Let me throw him out. Just a second. I'm very sorry. A little monkey had come in. My son had walked in and he wanted to see what we're doing. So, okay. So, the problem is when there is lots of ammonia, the backward reaction will become favorable and alpha ketoglutarate will get depleted. This alpha ketoglutarate, as you know, is a part of the Krebs cycle. Krebs cycle, also known as the TCA cycle, and the TC cycle is the main source of energy. It is generating maximum amount of ATP from the different metabolic pathways. All of them will converge at the TC cycle, and then all the ATP we get is from the TC cycle. But if you remove one of the components from the cycle will slow down. When the cycle will slow down, the ATP production will fall. So we have less amount of the ATP. The ATP production will fall. Because of this, we have the encephalopathy. So what are the questions like? Which substrate of Krebs cycle gets depleted in ammonia toxicity? Okay. What is the product which is formed in ammonia toxicity? Glutamate will be quite high. This glutamate can then be converted into glutamine. So glutamine will also be elevated. Okay. Common presentation. What is the common presentation? Common presentation is encephalopathy. Because of decreased availability of ATP, ATP is less, so we get the encephalopathy. So these are some questions which are asked. You should remember these points. Alpha ketoglutarate from the Krebs cycle. uh, because of this we get the encephalopathy. This happens because the reaction is reversible. A lot of glutamate will accumulate, which can then form the glutamine. Okay. So that was stage 2. Now this free ammonia is available. We have to take this ammonia to the liver. For transporting the ammonia, we attach it again to the glutamate. The enzyme involved is the glutamine synthetase. So what happens here? The glutamine is formed. So in all the tissues, this glutamine will be formed, and then this glutamine will travel to the liver. So the ammonia is transported by the action of glutamine. Okay. So question which is asked is, what is the form of ammonia transport? In which form the ammonia is transported from all the tissues? The answer is glutamine. But what you have to remember here, for muscle, the transport form is alanine. For muscle, the transport form is alanine. For all other tissues, the transport form will be glutamine. So remember, for muscle, the transport form is alanine. For all other tissues, the transport form of ammonia is glutamine. Very commonly repeated question. Very commonly repeated question. So we should know about this. Very commonly repeated question. You must know this. So now we get the glutamine. This glutamine will reach the liver. In the liver, glutamine is converted back to first glutamate, and glutamate is converted to back to alpha ketoglutarate. In both the reactions, the free ammonia is released. So now the ammonia from all over the body is now getting transported to the liver, where it is released. It's gotten stuck. Okay. Okay. So in the liver, the glutamine will get converted to glutamate. From glutamate, will get converted to alpha ketoglutarate, and in both the cases, we get the free ammonia. Now this ammonia is available to participate in the urea cycle reaction. It is available to participate in the urea cycle reactions. So let us now go on to the urea cycle reaction, which is the stage 4 in the metabolism of the amino group. Please note, urea cycle is also known as the Krebs-Henseleit cycle. Urea cycle also known as Krebs-Henseleit cycle. The two scientists which are working on this pathway. So about urea cycle, what are the points you have to remember? At the end, you are going to get urea, which is the degradation product of the amino acid metabolism in the humans. Synthesis occurs only in the liver, but here two compartments are involved. Both mitochondria and cytoplasm both are involved. Mitochondria and cytoplasm both are involved, and 4 moles of ATP are required. 4 moles of ATP are required. In addition to ammonia, carbon dioxide and nitrogen from aspartate. This question is asked, what are the donors of nitrogen? The donors of nitrogen are number 1 ammonia and number 2 the aspartate. Donors of nitrogen are ammonia and aspartate. How much ATP is required? Answer is 4 ATP. Which compartments involved in the synthesis? Answer is mitochondria and cytoplasm. There are 3 main questions which are asked. Okay. So what we can do, quickly take a look at the reactions, and then I'll list some of the disorders which are also associated with the urea cycle. Okay, so CO2 plus ammonia, both of them enter inside the mitochondria with the help of transporters, and then they are acted upon by the first enzyme, the carboxyl phosphate synthetase 1, commonly written as CPS1. Commonly, this is written as the CPS1, and you can see for this reaction we require N-acetyl glutamate. For this reaction, we require the N-acetyl glutamate, resulting in formation of the carbamoyl phosphate. Carbamoyl phosphate will be formed at the end of reaction 1. So what all to note? Enzyme is CPS1, N-acetyl glutamate is required, and 2 ATP are consumed. See here, the 2 ATP are getting used up. 2 ATP are getting used up, resulting in formation of carbamoyl phosphate. To this carbamoyl phosphate, the ornithine will come and get attached under the influence of the enzyme called ornithine transcarbamylase, written as OTC. Ornithine transcarbamylase, also written as OTC, will result in formation of the citrulline. Citrulline is formed, you can see here. Citrulline is formed, which is then transported across the membrane with the help of a dedicated transporter. You can see here the dedicated transport is there, which will bring out the citrulline. Citrulline will combine with aspartate. You can see the citrulline combines with aspartate, resulting in formation of the arginosuccinate. Okay, and in this process again you can see ATP converted into AMP. This time it is AMP. So we write it as 2 ATP equivalent. Whenever you get AMP, it implies that 2 ATP have been used up. Here the enzyme is the arginosuccinate synthetase. Arginosuccinate synthetase. Arginosuccinate is then quickly split into two. It is split into the arginine and the fumarate. It is split into arginine and the fumarate by the enzyme arginosuccinase, also known as the arginosuccinate lyase. Also known as arginosuccinate lyase. And lastly, the arginine is acted upon by the arginase, whereby we get the urea. Okay, so arginine will split into urea and the ornithine. Please note that this fumarate will enter again into the mitochondria. It will enter again into the mitochondria, and in the mitochondria, this fumarate first gets converted into the malate. Malate gets converted into oxaloacetate, and oxaloacetate is finally converted into the aspartate by transamination. We just discussed the transamination. So by transamination, the fumarate is finally converted into aspartate. This aspartate will go out from the mitochondria. It will go out from the mitochondria and again participate in reaction 3. So fumarate which is formed is getting recycled. The fumarate which is formed is getting recycled, and in this way the two pathways, the Urea cycle and the Krebs cycle, are connected. This is another question which is asked: which substrate will link the Urea cycle and the Krebs cycle? And the answer should be fumarate. If they ask amino acid, for amino acid the answer should be aspartate, but if they ask substrate, for substrate the answer will be fumarate. So you have to read the question carefully, what they are asking, whether it is fumarate or it is aspartate which has to mark. If they say substrate, answer is fumarate. If they say no, which is the amino acid which is linking the two, then it will be the aspartate. Okay, please note, for transporting the aspartate again we require a transporter. This transporter is commonly known as citrin. This transporter is commonly known as citrin. It is the aspartate glutamate transporter, commonly known as the citrin. Okay, so these are the different reactions which are involved and the different pathways, so different proteins which are involved. So here I would like to mention one more thing, that for the synthesis of N-acetyl glutamate we have a very specific enzyme called N-acetyl glutamate synthase. N-acetyl glutamate synthase, which will synthesize the N-acetyl glutamate. Why I'm telling you this? Sometimes this enzyme may be deficient, giving rise to one of the important clinical conditions. In addition to the other enzymes which I already mentioned, other enzyme means the CPS1, ornithine transcarbamylase, the arginosuccinate synthetase, arginosuccinate lyase, the arginase, and the transporters. The transporter for the ornithine and the transporter for the aspartate. These are the important molecules which can cause the clinical conditions. So let's take a quick look. Metabolic disorders which are associated with the reactions of urea cycle. Okay, so first is the hyperammonemia type 1, which is due to deficiency of the first enzyme which we had said, carbamoyl phosphate synthetase 1. Deficiency of CPS1 will cause hyperammonemia type 1. Hyperammonemia type 2 is due to deficiency of the ornithine transcarbamylase. Due to deficiency of ornithine transcarbamylase. Then we have the citrullinemia type 1 due to deficiency of the third enzyme, that is arginosuccinate synthetase. Deficiency of fourth enzyme causes arginosuccinic aciduria. Arginosuccinic acid will accumulate. If it cannot be broken down, then it will pass off in the urine, causing the arginosuccinic aciduria. Lastly, if the arginine cannot be broken down by the arginase, if arginase is less in amount, then the arginine will again start accumulating, causing the hyperargininemia. So here the deficient enzyme would be the arginase. If there is NAGS deficiency, I told you sometimes in some individuals the NAG synthase may be deficient. So here the presentation is similar to the type 1, that is hyperammonemia type 1. It will be similar. Why? Because the first reaction only will get blocked, whether the enzyme is not there or the coenzyme is not there. The end result is the reaction 1 will not happen. Okay, so similar presentation will be there. if there is problem in the transport of aspartate, I told you the transporter is the aspartate glutamate transporter, then we get the citrullinemia type 2. Why? Because aspartate will not be available to combine with the citrulline which has been formed. So the citrulline will start accumulating and it will cause the citrullinemia type 2. Lastly, we have what is known as the triple H syndrome. In triple H syndrome, we have the hyperammonemia, hyperornithinemia, and this ornithine gets converted into the homocitrulline. So we get the homocitrullinemia. We get the homocitrullinemia. Okay. So the 8 disorders associated with the urea cycle, these are the metabolic disorders urea cycle, and part of what we call IEM. I mentioned this in the group today also in the Telegram group. IEM, which stands for inborn errors of metabolism. So urea cycle defects are a part of the cluster called inborn errors of metabolism. Okay, these 8 would be a part of what is known as inborn errors of metabolism. Please note, inheritance pattern is autosomal recessive. Inheritance pattern is autosomal recessive, except for hyperammonemia type 2, which is X-linked, and a common feature is orotic aciduria. Common feature in urea cycle disorders is the orotic aciduria. Now, in the approach to management, the most important thing we have to note are two molecules which increase the excretion. First is benzoate, commonly given as sodium benzoate. So benzoate will combine with glycine and it will form the hippurate, and you know this hippurate will be lost in the urine because it is easily removed. It is water soluble and it is filtered, it is secreted, and it is quickly removed. Second is phenyl acetate. Now we don't give the phenyl acetate, rather we give the phenyl butyrate. So phenyl butyrate is the prodrug. Please remember, if the question is asked on prodrug, so phenyl butyrate is also a prodrug. This phenyl butyrate will get converted into phenyl acetate. The phenyl acetate will combine with glutamine to give you the extremely water soluble complex phenyl acetyl glutamine. It will get filtered, but we don't have a transporter for phenyl acetyl glutamine, so it cannot be reabsorbed. Okay, it cannot be reabsorbed. So please remember these two names. Sodium benzoate will result in formation of hippurate. The phenyl butyrate, which is a prodrug and gets converted into phenyl acetate, will finally form the phenyl acetyl glutamine, which will again be lost in the urine, just like in the first case. It will be lost in the urine, but net result is we are removing the nitrogen from the body, so the ammonia levels will also start falling and the patient will improve. Please remember this. In addition to this, sometimes you can do the supplementation. Supplementation, for example, if the arginine is not being formed, enzyme number 3, arginosuccinate synthetase is not there, or arginosuccinate lyase is not there, then the arginine will not be formed. We can give the arginine from outside. And lastly, the N-carbamyl glutamate can replace the N-acetyl glutamate. So if there is deficiency of the N-acetyl glutamate synthase, then acetyl glutamate will not be formed. There you can give the supplementation of N-carbamyl glutamate. So in this case, what we do, we try to bypass the block. Wherever the block is there, we try to bypass it by giving the suitable supplements, so which are arginine and the N-carbamyl glutamate. So these are the urea cycle disorders. A lot of questions we get from urea cycle disorders. You can get the question in the form of the clinical case also, and you should be able to identify them. Main role in management for these two molecules, sodium benzoate and the phenyl butyrate, which gets converted into the phenyl acetate. So this was about the metabolism of the amino group. Now what we are going to do, we are going to look at the breakdown of one another important amino acid which is associated with multiple inborn errors of metabolism. So we are going to look at the second cluster of inborn error of metabolism. Let's look at the second cluster of inborn error of metabolism, which starts with the metabolism of phenylalanine. Phenylalanine normally converted to tyrosine by the enzyme phenylalanine hydroxylase. In some individuals, when there is deficiency of the enzyme phenylalanine hydroxylase, it will give rise to the condition called phenylketonuria. It will give rise to the condition called the phenylketonuria. But important point to remember here is this enzyme also requires the coenzyme, which is the tetrahydrobiopterin. In due course, it gets converted to dihydrobiopterin and then it is recycled to reform the tetrahydrobiopterin, which can again participate in the reaction. So not only the enzyme deficiency, we can have the coenzyme deficiency also. Okay, and then also you'll get the phenylketonuria. What all do you have to remember for phenylketonuria? Number one, it will present with mental retardation. Okay, number one, it will present with mental retardation. Number two, you will have mousy or musty odor. Mousy or musty odor, which is due to phenyl acetate, which is due to the phenyl acetate. So this is being asked in multiple ways again and again, so I'll try to tell you how they'll try to confuse you. They will say mousy musty odor present in PKU because of phenyl lactate. Wrong. They will say mousy musty odor is present in the patient of PKU because the tyrosine is not there. Again, wrong. Why the mousy musty odor is there? It is because of the presence of the phenyl acetate. And where can you find this mousy or musty odor? You can find it in urine and in the sweat. You can find it in the urine and in the sweat. Okay. Second thing which you have to remember in this case, in case of PKU, is that this is one of the condition for which you have the neonatal screening. Why? The incidence is not rare and the mental retardation which occurs is irreversible. So you need to catch a mental retardation immediately at birth and institute the management. So last part is, what is the management for these patients? The answer is very very simple. The answer is dietary restriction. You don't need to do anything else. You have to give the dietary restriction, but this is a challenge because phenylalanine free diet is not prepared in our body. In our country, it has to be imported from outside. So a lot of import restrictions are there in the import of these. So you have to find suitable supplements which are free of the phenylalanine. Okay, suitable supplements which are free of phenylalanine. In the normal course, the tyrosine is then converted into the hydroxyphenyl pyruvate by the enzyme tyrosine amino transferase. The tyrosine amino transferase will convert tyrosine into hydroxyphenyl pyruvate. If there is deficiency, of the enzyme tyrosine aminotransferase, we get the condition known as tyrosinemia type 2, also known as the Richner-Hanhart syndrome. So if you see the term Richner-Hanhart syndrome, immediately you have to think, okay, we are now talking about the tyrosinemia type 2. Sometimes the tyrosinemia type 2 is also known as oculocutaneous tyrosinemia, because these are the two places where commonly we see the sign and symptom. Okay, so in the eye we find the keratitis, in the palms and soles we find the blistering lesions. So oculo and cutaneous, eye and the skin are getting involved, so we call it the oculocutaneous tyrosinemia. We call it the oculocutaneous tyrosinemia, which is the other name for tyrosinemia type 2. Now, in the course of normal metabolism, the tyrosine is getting converted to hydroxyphenyl pyruvate, which is subsequently converted into the homogeneity by the enzyme hydroxyphenyl pyruvate oxidase. Now, here there are two things. Number one, when there is deficiency of the enzyme. When the enzyme is deficient, we get the neonatal tyrosinemia, also called tyrosinemia type 3, which is mostly asymptomatic. In most of the patients, there will be no symptoms, but in some cases, you can get the mental retardation, which is feature of tyrosinemia. In tyrosinemia, we commonly find the mental retardation, but most of the type 3 patients are asymptomatic, so we don't have any mental retardation. This is the neonatal tyrosinemia. On the other hand, in some individuals, the enzyme is there, but it is not working properly. We call it a defective hydroxyphenyl pyruvate oxidase. And in this case, when the defective enzyme is there, we get the product called Hawkinson. An intermediate product is formed. The metabolism will stop somewhere in between. An intermediate product is formed, which we call the Hawkinson. Because this Hawkinson can come out in the urine, the clinical condition is called Hawkinsinuria. Clinical condition is called Hawkinsinuria, where we have the chlorine like odor in the urine. Chlorine like odor in the urine in case of Hawkinsinuria. Please keep note of the enzyme. Hydroxyl pyruvate oxidase is defective. It is not decreased, but it is defective. If the enzyme is decreased, then we get the tyrosinemia type 3. I already told you, tyrosinemia type 3. In the normal course, the homogentisate is getting converted into fumarate acetate by the enzyme homogentisate oxidase. Very, very commonly repeated question. In some individuals, again, this enzyme may be deficient. When the deficiency is there, we get the condition called alkaptonuria. The classical feature in alkaptonuria is blackening of urine on standing. If you leave the urine in a tube exposed to the air, after some time it will acquire the black color. So blackening of urine on standing is the characteristic pathognomonic feature of the alkaptonuria. In addition to this, you should note that patients may present later. Later means at the age of 30s and 40s with back pain. Why back pain? Because the damage is happening and the whole IVD has fused due to fibrosis. They will have the arthritis. They will have early onset arthritis. Okay. And the last very commonly asked question is, they will have what is known as ochronosis. What do you mean by ochronosis? Ochronosis is black pigmentation of the skin. On the skin, the black pigmentation will appear. That is ochronosis. This is the extrapolation of blackening of urine on standing. In atmosphere, oxygen is high, so the oxidation occurs very fast. But in the body, the oxidation will occur low because partial pressure of oxygen is much lower. That is why the ochronosis appears in the third or the fourth decade of life, 30s and 40s. That is when the ochronosis will become apparent. Lastly, the conversion of fumarate acetate to fumarate and acetoacetate, where the enzyme fumarate acetoacetate hydrolase is required. If there is a deficiency in the enzyme fumarate acetoacetate hydrolase, we get the tyrosinemia type 1, also known as tyrosinosis. Sometimes also known as the hepatorenal tyrosinemia. So in hepatorenal tyrosinemia, the name itself tells you that the liver and kidney both are getting affected. So sometimes in these patients you find the rickets. Why? Because the kidney is not able to retain the calcium, and the rickets will occur in these patients. Please note, in the tyrosinemia type 1, we will obviously have the mental retardation. We have the mental retardation, and you can also have the renal stones. Nephrolithiasis can be there in the tyrosinemia type 1, and obviously the tyrosine levels are significantly high. Tyrosine levels are significantly high, whether you're measuring it in the blood, whether you're measuring it in the urine, the tyrosine levels will be high in tyrosinosis type 1. Okay, we talked about these. Please note, for tyrosine type 1, for treatment, we have the drug anti-BC or nitisinone. This drug can also be used for another condition. Can you tell me what is that condition? It can also be used for management of the alkaptonuria. Okay, it can also be used for management of alkaptonuria. Okay. The last point which I want to mention here is a condition called MSUD. MSUD stands for, I'm sure all of you know this, MSUD stands for the maple syrup urine disease. As you can make out, the maple syrup like nature of urine is there, but in what terms? Does it taste like maple syrup? We don't know. We've not tasted it. Then why we call it maple syrup urine disease? The odor is like maple syrup, sometimes also described as the burnt sugar odor. Okay, so maple syrup urine disease. If you look at the problem is branch chain keto acid dehydrogenase deficiency. The branch chain keto acid dehydrogenase deficiency. Now remember, which are the branch chain amino acids? The branch chain amino acids are the leucine, isoleucine, valine. That is why their levels will be elevated. Their metabolites, alpha keto acid, alpha hydroxy acid, they will also be elevated. But after the formation of alpha keto acid, you can see here the keto acid dehydrogenase deficiency is there. Further metabolism is not happening, so the accumulation will take place. Inheritance pattern is autosomal recessive for the maple syrup urine disease. Inheritance pattern is autosomal recessive. One of the diastereomers of isoleucine, alloisoleucine, are found. And last, the neurotoxic substances are leucine and 2-isocaproic acid. Leucine and 2-isocaproic acid. Till now, we are talking about the defect in the metabolism of the amino acid, but sometimes the defect can be there in the transport. The problem can be there in the amino acid transport. First example is cystinuria. So here, what you have to remember: the amino acid lost in the urine are cola, cysteine, not cysteine, cysteine, ornithine, lysine, and arginine. Cysteine, ornithine, lysine, and arginine. They are present in urine in cystinuria. Second condition is the Hartnups. Second condition is the Hartnups. In Hartnups, there is defect in the transport of large neutral amino acids and aromatic amino acids. Okay, so one of those amino acids is the tryptophan. So tryptophan will get lost in the urine, it will not get reabsorbed. So it will manifest as tryptophan deficiency. Remember, tryptophan gives rise to niacin, that is vitamin B3. Okay, so it will present as niacin deficiency, which I'm sure all of you know will cause what is known as pellagra. So we see a patient having the pellagra, and if the small child, then we start thinking about the differential of defect in the amino acid transport. Inheritance pattern, autosomal recessive. Inheritance pattern is autosomal recessive. Okay. Okay, let's take a quick look at some of the questions. The transfer of amino group from amino acid to an alpha keto acid is done by, we just mentioned, the reaction is transamination. The name of the enzyme is the transaminase. Okay. The amino acid which serves as a carrier of ammonia from skeletal muscle to liver. Please note, it is from skeletal muscle to liver, not from rest of the body. So glutamine will not be the answer. The answer will be alanine. The answer will be alanine. Which product of citric acid cycle is used in detoxification of ammonia in the brain? We just discussed this. The answer is alpha ketoglutarate. The answer is alpha ketoglutarate. Then the question is, urea cycle occurs in which compartment in the body? Urea cycle occurs in which compartment in the body? GIT, spleen, kidney, or liver? I just told you urea cycle where it is happening. Urea cycle is happening in the liver. Urea cycle is happening only in the liver. Then this question I told you is, our source of nitrogen in urea cycle are. One of them is ammonia. So here also ammonia, here also ammonia, here also ammonia. Second is aspartate. So the correct answer becomes aspartate and ammonia. The two sources of nitrogen in urea cycle is aspartate and ammonia. The carbon will come from respiratory CO2. Which of the following is an urea cycle enzyme? See, glutaminase is not related to urea cycle. Asparaginase is not related to urea cycle. Glutamate dehydrogenase, okay, you can think of it, but not directly. Directly in the urea cycle, we have arginosuccinate synthetase, which will make arginine from the citrulline and aspartate. Correct answer is arginosuccinate synthetase. At physiological pH, what is the nature of carboxy terminal of the peptide? Just remember, at any pH greater than 3, the carboxy terminal will be negatively charged. At any pH greater than 3, the carboxy terminal will be negatively charged. So here we are talking about the substitution. When you do the substitution, in that case, similar type amino acid have to be added. So remember, isoleucine is a branch chain amino acid. Which are the other branch chain amino acids? Valine and leucine. So if you want to replace, if you want to do the substitution, you have to put the similar ones. So you have to look for valine and leucine. Valine, yes, valine. So the correct answer should be valine. Correct answer should be valine. Amino acid having amino side chain. We had marked this answer. The answer is proline. Amino acid having the amino side chain. Answer is proline. Basic amino acid are all except, remember the mnemonic HLA, HLA, HLA. HLA means histidine, lysine, arginine. Proline is not a basic amino acid. Non-essential amino acid. Non-essential amino acid means which can be synthesized in our body. And you know, tyrosine can be synthesized from the phenylalanine. Okay, so tyrosine is non-essential, whereas phenylalanine will be essential. It must be provided in the diet. It cannot be synthesized in our body. Amino acid for which dissociation constant is closest to physiological pH, the answer is histidine. Please note, the histidine will have the imidazole side chain. It will have the imidazole side chain, which has a dissociation constant close to physiological pH. Amino acids secreted in cystinuria are, if you remember, cola. Cysteine, not here. O for ornithine, L for lysine, A for arginine. Means histidine is not uh, being lost in the cystinuria. Which of the following is not synthesized from tyrosine? When you talk about tyrosine, it will give you three things. It will give you the catecholamines. Okay, tyrosine will give you the catecholamines. It will get converted into melanin. It will get converted into melanin, and it will give rise to the thyroxine hormones. When I say thyroxine hormones, it means both T4 and T3. Okay, so norepinephrine is a catecholamine. Thyroxine is a catecholamine, and in the process dopamine is also formed, which is a catecholamine. So please note, melatonin is not formed, rather melanin is formed. If it is melanin, it will be correct, but it is melatonin. So melatonin is not synthesized from tyrosine, rather melatonin will come from tryptophan. It will come from tryptophan. Tryptophan also gives rise to the serotonin. serotonin. And you already mentioned the tryptophan will give rise to niacin, that is vitamin B3. So there are different sources, different products coming out from the tryptophan. Which amino acid does not show post-translation modification? Just told you one of the amino acid getting inserted during the translation. Here we have the co-translational insertion. This amino acid is added during the translation. For others, they are modified amino acid. You do the modification after the peptide chain has been formed. In phenylketonuria, the main aim for first line therapy. First, you have to understand what is the first line therapy. First line therapy is dietary restriction. Why do you want to do the dietary restriction? To bring down the phenylalanine. When you bring down the phenylalanine, the alternate metabolites which are harmful will not be formed, so it reduces the substrate for the enzyme. The mode of action of phenylbutyrate in urea cycle is by, you said it will remove the nitrogen by alternate route. It will remove the nitrogen by alternate route. It will form the phenylacetyl glutamine, which will be excreted in the urine. In this case, it is getting excreted via the urine. Okay. Let's take a short 5-minute break and then continue with the topic of protein. There's just a washroom break. I've got some separate TM. So let's take a short 5-minute break and then we'll start.